University of Latvia. But I work in the Latvian Biomedical Research and Study Center, and this is where this study was done. So today I present you a novel phage isolated from the wastewater of Latvian city Jelgava, named Jelgavas-1, and that was found to infect a strain of AEROMONAS salmonicida, subspecies salmonicida, and it was identified by partial 16S rRNA gene sequencing, and also found in the same wastewater sample as this phage. So, what is AEROMONAS salmonicida? It is a common pathogen that causes furunculosis in fish, leading to antibiotic overuse and antibiotic resistant strains emergence in aquacultures. So this bacterium infecting phage was isolated and propagated. There you can see the plaques that it made and podoviral morphology variants in TEM microscopy. Then Illumina platform compatible next generation sequencing library was prepared from this phage genomic DNA, and it was sequenced on Illumina's MISEQ system. Obtained reads were assembled into a 40,505 base pairs long genome, and it was validated in a wet lab by restriction analysis that you can see here, and a combination of comparative genomic approaches was utilized to functionally annotate this phage genome. And here you can see 53 open reading frames and 28 products with annotations, and they are marked by color, by functions. There you can take a closer look later. And as we can see here, from the 9 most closely related phages, there's only up to 40% similarity in already published ones, which indicates the importance of this phage novelty. And also, as you can see here, major capsid protein 3 is it's actually more similar to Vibrio phages than already existing AEROMONAS phages. And here, the RNA polymerase 3, it's also a very low similarity between other AEROMONAS phages. So, do the microbiology experiments. So I made adsorption test, and this phage actually shows pretty good adsorption capacity and time, as you can see here. And I made also a one-step growth curve, and the latent period of this phage is pretty low. It's about 33 minutes, and the burst size is 86 phage forming units per cell. And this phage also shows great ability to effectively lyse its host in vitro, even at low added phage concentrations, even at low multiplicities of infection, as you can see here. And this phage is pretty stable also at pH from 5 to 9, and even at 60 degrees, 30-minute incubation, it still shows viable plaques. And here I made some stability experiments, defrosting multiple times in different types of medium, and also I defrosted them after half a year. And there you can see the results. So in conclusion, this phage represents not only a novel phage species, but also a novel possible phage genus in the family of Autographviridae. And so this strictly lytic phage does not harbor any genes that might enhance the fitness or virulence of its host. Therefore, it seems to be a more suitable candidate for studies evaluating the phage biocontrol possibilities of AEROMONAS salmonicida in an aquaculture setting. So yes, that's all from me. I hope that if it interests you, you can maybe write to me in this email, and we can chat, or here is the email, and maybe do some collaboration work. Thank you for your
Thank you very much, Karina. So we have one question, first of all, from Yulia Payuk.
Okay.
Yes, do you hear me?
Yes.
Okay, so thanks, Karina, for your nice presentation. Nice to see work from Nikita's lab. I have a question. What about RBP, receptor binding protein? Does it have any depolymerase activity, or it's just the same GP17 as in T7, but it lacks some similarity in C terminus?
So I think it actually binds to lipopolysaccharides, as this bacteria is gram-negative, and as we can see here, it's the coverage with spike protein was very low to already sequenced ones, so it's pretty unique, and I think it has depolymerase activity.
Okay, so you have not only the tail fiber, but also the spike.
The tail fiber and the tubular protein A and B.
But tail tubular proteins A and B, they are generally in the tube. Do you have tail fiber? Where do you have tail fiber?
There are tail fibers.
Okay.
And they are very low.
Low similarity.
Low similarity between these phages. No, actually, I don't have spike protein. I have tubular and tail fiber. Yeah.
Mm-hmm. Okay, so generally, I think it just have some different receptor binding activity. Okay, and why do you state that this is a new genus? Have you performed the whole genome comparison against the closest
Yeah, this is the closest relative. This is the closest relative, the AEROMONAS phage T7AH. And these are the 9 most closely related phages, and the similarity is if it's not 75%, then it's considered a putative new phage genus.
But here you have similarity 100% with AEROMONAS phage.
This is my phage.
Ah, this is your phage.
Yeah.
Okay. It don't represent it like this.
Okay, thank you for
yeah, okay, so this low similarity 72, yeah, I think it can be the new.
Yeah, it's calculated by multiplying the coverage and identity. So this by this is this.
Okay. Okay, thank you.
Good job. One question from our co-chairman of conference, Larissa Spika.
I have a little question. Please comment and tell a couple of words about the mode of therapeutic using. It will be topical using?
The use could be very varied. It can be actually injected in vivo. It can be added just in a fish tank, or maybe the fish could be submerged into this phage and then released. There are many possibilities that we will hopefully study next in vivo.
Thank you.
Thank you so much for your amazing course, Svan.